We're going to do this together. So now we have a split screen of your spreadsheet on the right. We're going to use it as a calculator. We're going to do some graphing in the spreadsheet, but we're following closely the directions of continue and save, and so we're going to go back and forth between the two. So we've set this up with a split screen. So we are on module 4 once again, and when you click on module 4, it's got this opening video that we just saw, so I'm going to click continue. So the video explained the dewlap appearance is important because it is a mate selecting reproductive isolation technique for each species. So we're going to click, and I'm going to minimize the spreadsheet so that I can see the picture of the Anole, and I'll explain what we're going to do. This is our data collection. We're going to be presented with 10 different anoles, and we're going to assign a color to, no, we're going to assign a number to the color. So I'm going to click over here to the hypothesis. I could have done this first to give you an idea of what we're doing here in the lab. So the question of the lab is, can dewlaps really lead to speciation? Is it something that is statistically shown to be a difference? Or are we just assigning it like, hey, they look different colors. Does it really cause them to be reproductively isolated? They're in the same spot, but they have different colors of dewlaps. So what is the independent variable that we are testing here? It's the color of the dewlap. So over here in hypothesis, the independent variable, the one that we're testing, as you can see, we're going to have some that have a light dewlap and some that have a dark dewlap. So we don't know what species they're in, but we do recognize that they're different colors. So the independent variable is the color of the dewlap. What are we going to measure? It's right here on the screen. We're going to assign a number to the color. 6 is going to be bright and 1 is going to be dark. And so we're going to have a sliding scale. So what you're going to do now is slide these colors so that it matches. I'm not sure if yours shows up in the same order, if it's random. So don't just go off of my numbers. I'll get you started, then you have to do it on your own screen, because it doesn't really, it does say 1 of 10 measurements. So I'll slide this one over. I think it might be a 3. Maybe you would see it as a 2. I kind of see it as a 3. It's a little lighter at the bottom, and it's right at the tip of the arrow. So right there where the arrow is, I think it might be a 5 or a 6. Maybe it's, I'll go with 5. If you're zoomed in too far, you can't see the word continue. So make sure you're zoomed out, and so that you can see the whole screen while we're doing a split screen. Then I'm going to click next. I'm on number 2. So go ahead at your own speed and see if you can now give a color. Give a number for the color at the top of the dewlap and the bottom of the dewlap, and then it is going to calculate the average between the two numbers. And it's going to keep track of your data for you. So I just want to see if you're on the right screen to be able to do this so that you could go through it. Yep. It is the number we're assigning to the color.
Wait, what's the number?
You should have the spot where. So we're going to finish the hypothesis. The hypothesis is always written with the 2 variables. You in the spot where you can analyze the dewlaps? There you go. Analyzing spot. So you're back here. Can you open up the spreadsheet? Did you have it open earlier? Yeah, the one that we did the questions. Yeah. say click first, directions, then up at the top, it's straight to the website. We have the website link. And you can do a split screen. We have to learn lots of different forms. Good. You're analyzing. You're all done. So before we leave the hypothesis, let's do that real quick. The formula is always if ... then. And it's if independent variable, then dependent variable. So we said the independent variable was color. So if we examine the color variation of dewlaps, then we will see that they vary by numbers that we can assign to different species. If we examine the color, that's the independent variable, then we can assign a number to that, which will show us the statistics of why they're reproductively isolated, forming two different species. So it sounds complicated, but it's if independent variable, then dependent variable. So whatever you wrote down for these two, you just say it if I examine the color variations of their dewlaps, then I can see a statistics, that means a number value, which we can then use to evaluate how they're different species. So go ahead and you've got to do your 10 so that you can get to a chart. Angel, are you with us?
Yeah.
Remember what I said at the beginning? Now I did this earlier and I didn't start over, so I'm going to delete this to kind of show you where I got the numbers. So the important thing isn't just following my numbers, but knowing how to get your own numbers, which is going to be based on how you analyze the colors. So if you look over here under our spreadsheet for data, once you are at this, after you've analyzed all 10, 5 of each of the species, we have to do calculations. So you can open up a calculator here, pull out your Texas Instrument, but know that a spreadsheet is a calculator. Every cell of a spreadsheet, it can do math. And we've done this throughout the whole year. So I don't know if you have realized the method to my madness of showing you spreadsheets, because it will help you in high school, college, and in life if you know how to do spreadsheets. It's invaluable for you to be able to analyze data. So what you need to do now is, the directions up here say in the yellow shaded area, oh, my screen is pausing. In the yellow shaded area, enter your mean dewlap colors. See these colors that you have right here? This is a Christatelis. So these numbers I'm going to put into the spreadsheet in the yellow area. Whatever your numbers are, don't go off of my numbers. They may or may not be the same. 4. You're taking the mean color between the top and the bottom of the dewlap, and the website calculated the mean for you. And now the directions over here at the spreadsheet say now take the blue shaded areas where I built out the formulas for you. You could have done the same thing yourself. So if you want, tap into it later and see what the formula is. And I need to take these four numbers that you see down here, the mean of all of the samples. The standard deviation tells you how close to the average your numbers are. Like if this was a high number, like 10, that means that, wow, you really had an outlier, like a really dark one and a really light one. And so the numbers are all over the board. That would almost be like a test where you had A's and F's and no numbers in between. The standard deviation on that test population would be a really high number. So you want a really small number, really with standard deviation. That means that the numbers don't vary much from the mean. And now do you see why I have this skull right here? And it says, at first I said numbers to blow your mind because some of you would go numbers and it, like I'm done. Like my brain can't hold that. So you can take a look at it. It's I even Googled it. I said, define this in layman's terms because sometimes it's like too much. So the mean is the average, the standard deviation is how much the number varies from the average, and then you get into a standard error of the mean, which tells you, interesting chart right here. If we have one sample and then we calculate the standard error, that means that this one sample can tell us about all of the population. Even though we're only doing 10, it can tell us what 1,000 organisms are if that number is within line. So the standard deviation tells you how close one class is, but since I have 5 classes, if I did the standard error of the mean with your class, it would tell me how every class would do. So it's interesting. So there's a lot of statistics. That's why those guys were in the field measuring and doing all this particular stuff so they could go back to the lab and analyze it. So now you're going to take these blue numbers that you see in the chart and you have to enter them right over here. So I'm going to do that. It's 5.5 for that guy. It's 0.856. Go off of your numbers that you have. 0.387, 0.775, 3.9, and now you have to be zoomed out far enough so that you can see the word save and continue. When you save and continue, you now see your data summarized right there, and the scientists in the field is right here. So how close did you get? We're going to go off of both of this. We're going to compare, and we're going to do work with both sets of data now. So notice that yours and theirs is going to pop up. You need to take a screenshot of that. If you look at the directions at the spreadsheet, it says do a screenshot, and you're going to put it underneath the picture tab. So now when you go to your spreadsheet and click on pictures, there's what the picture looks like without the numbers. You need to tap on that, save this screenshot, and insert your picture into there. So you can tap on this picture now and delete it and insert your own picture in there. So I'm going to come around the room again and see how you're doing. Very good. So you're farther ahead. Yeah, you grab it and you raise the bar up to the height of your mean. So you need to go back and click on results. And then save and continue. And it's right there. So take a screenshot of that. Good. Anybody need help? Looks like you're doing good. All right, so now you get the technical aspect of what we're doing here. We're using our spreadsheet to summarize all the data. The website is calculating part of it. We're using the spreadsheet as a calculator and taking some of the info back. And now we're going to graph it. So we're graphing the results. And the way you do that is you grab the top of the bar and with your finger you slide the bar up and down.
Do you want us to compare the two graphs or just use one graph?
And because I did this earlier, there we go. I'm able to move it around. So what you're going to do is you're going to use your data, which is right over here. So the standard mean is 3.9. So I'm going to go over here and slide this up to 3.9. It even tells you what number it's got, so you can be dead accurate with it. And then that bar is called the standard error bar, and you drag it just like it shows, where half the bar is above and half of the bar is below it. So if that is compressed, that means that the number can be anywhere in between here, and that's 95% certain that you have the mean. So we're dead even right in the middle of our 95% confidence. So this is a good statistical measure. So then you click Save and Continue, and you compare your results. And this is the second picture that you need. So here's your second screenshot. You don't have to be dead accurate with the scientists, but the A. cristatellus species should be much higher than the A. cookie species. So if you're off, like mine was 5.6 and theirs is 4.8, that's because I judged the color slider bar slightly different than they did. They probably have a better scale than trying to see it on this computer screen. So it's pretty good though. Now do a screenshot and drop that right over there in the second picture. Click on the results or results graph. Oh, that's a good point. I went too fast actually. So do you see yours says title and it says x-axis and y-axis. I've already done that. So if you need to go back, you will click on the results or the results graph. So let me show you, because the directions say you have to have a title for your graph. So here's how you do that. When you are at your graph, you have to tap into the title and you have to tap into the x and the y axis and you have to name them. If you don't do that, you're going to lose just 1 point. So you should never turn in a graph that doesn't have a title and an x and a y axis. Otherwise, the chart is meaningless. So you will just tap into it and then give it a name. So a good name would be comparing two species dewlap colors. So the X are that's species, and the Y is the dewlap color average. The dewlap color mean, same thing. So if you need to go back, you would click on results, tap through a couple screens and get back over here and then go ahead and do the X, Y, and the title. Please don't forget that. So when you turn this in, the first thing I'm going to look at, do you have a title and do you have and so you can see that your title is going to be really close to their title.
Yeah.
You tap on it and slide it so that half of the line goes above the bar. And you get to there. So just continue. You tap on it. Sometimes and it needs to be like average. 5.2, that's great. 5.2. Error bar. Goes above and below. Good over here?
Do you answer these questions?
Yes. So we're done with our data, and you will click continue, and you've got questions to go through. There's 5 questions at the website, and then you'll go over to the spreadsheet as well, do these side by side, and you will see that it even says, answer in the website conclusion question 1. Which Anole species seems to have a brighter dewlap? And you look at your data. You can open the reference graph right there so that you don't have to lose your place. And it is the brightest one. The brightest was the highest number, right? Yep. So that is the Christatelis. So you click on that. It'll even tell you if you have the right answers, so then you know the answer to put in right there. So yeah, I skipped number 4 over here, and then there's a couple more questions for you to answer. So that's your lab write-up. And then when you're finished, you click on the 3 dots, export this as a PDF file, save it as a single page for each of the tabs, and away it goes as a PDF file, and you're going to be able to turn that in. You can send it to Notability or straight to Google if you want, and so that's it. This is due on Sunday night after vacation is over, so it's the 24th. But I think you're really close to finishing, right? Maybe you have to go through some questions.